everybody, and welcome to another episode of the Molecular Cell Biology YouTube channel. Today we will be talking about one method that is widely used to study translational rates associated to specific messenger RNAs, and that is the use of polysomal fractionation by using sucrose gradients. And before jumping into this, it's important to remember that translational regulation is an important mechanism to regulate gene expression in eukaryotic cells, specifically mammalian cells. So when you have a population of messenger RNAs in the cell, so here I'm drawing a messenger RNA, we have the poly A tail, we have the cap at the 5' end of the messenger RNA. So when you have a population of different messenger RNAs in the cell, different messenger RNAs can be translated at different rates within the cell. So then the question is, how do you go about identifying the messenger RNAs that are being translated at very high rates as opposed to those that are being translated at low rates? Well, in order to answer that, one key idea is that translation requires the association of the messenger RNA with ribosomes. So a messenger RNA that is being translated will have ribosomes associated to it. And the efficiency of the process of translation will depend to some degree on how many ribosomes are associated to that particular messenger RNA molecule. So the more ribosomes are associated to a single messenger RNA molecule, the more efficiently that particular messenger RNA will be translated. So again, more ribosomes means increased translation. And another key idea is that messenger RNAs that have ribosomes associated to it behave as if they were larger complexes. So having only one ribosome associated to a messenger RNA is different from having many ribosomes associated to it. And you can actually separate these messenger RNAs based on how many ribosomes are associated to a single molecule of messenger RNA. And that is what is accomplished using sucrose gradients. So you can actually separate messenger RNAs from each other based on how many ribosomes are associated to it. Now, another important idea is how many ribosomes can actually be fitted into a single messenger RNA molecule. And for that, it's important to keep in mind that one ribosome associated to messenger RNA will basically occupy about anywhere between 28 to 30 nucleotides in the messenger RNA. So one ribosome equals about 28 to 30 nucleotides. And there is some spacing that is required between adjacent ribosomes. And this is so that you don't end up having situations where one ribosome collides with another ribosome, because this will be bad for translation. If you have colliding ribosomes, you can easily imagine how one ribosome can actually push the other one, therefore forcing it to skip a few nucleotides, therefore affecting the reading frame that is being used for translation. So there needs to be some spacing between consecutive ribosomes, and that spacing actually can fluctuate. There are regions in messenger RNA molecules where ribosomes will move relatively smoothly and at a relative high speed. And in those regions of messenger RNA, the maximum packing that you can have is somewhere between 40 to 7 nucleotides of spacing in between consecutive ribosomes. So that's kind of like the normal spacing. But if you're going through a region that is difficult to translate in a given messenger RNA, that may actually lead to a slowdown in the progression on the movement of one particular ribosome, so that the ribosome that is coming behind may actually catch up and get a little bit closer to the other one. So in general, the spacing will be somewhere in this range, 40 to 70 nucleotides, but that can actually be affected by other factors, such as the use of rare codons in the messenger RNA, which may actually delay the movement of ribosomes and things like that. So in general, a good rule of thumb will be that you will have about one ribosome for every about 100 nucleotides in the open ribosome frame, in the coding sequence. So a coding sequence that spans for about 300 nucleotides will have about a maximum of about 3 ribosomes associated to it. Which also goes with the idea that the longer the ribosome frame, the more ribosomes you can actually have associated to a single messenger RNA. So those are things that may actually affect this analysis, because if you're looking for a messenger RNA molecule with a very long ribosome frame, then chances are the odds of finding that messenger RNA among the population of messenger RNAs that are loaded with more ribosomes is actually higher. So you have a better odds of finding that messenger RNA among the well-translated messenger RNAs simply because it has a very long open ribosome frame. So keeping that in mind, the way that you can actually tell whether a messenger RNA is well-translated or not is by doing these sucrose gradients. Okay, so the way that this works is you prepare first a sucrose gradient, and this is done simply by loading solutions containing different concentrations of sucrose in a centrifuge tube. So the denser the solution is, that is, the higher the concentration of sucrose present in a given fraction, the lower it will go in the tube. So you load the tubes from the highest concentration of sucrose, which will go at the bottom, to the lightest, the one that contains the lowest amount of sucrose. And then after that, you allow the tube to sit steady overnight, and that allows some diffusion of those gradients. So you end up with a linear gradient that spans the entire range of the different concentrations that were used to load on the tube. So now you have a continuous gradient from the lowest concentration up to the highest concentration. And then you take that tube and you load then the cell extract on top of the gradient, the cell extract containing translating messenger RNAs that had been stopped by using specific chemical inhibitors of the translocation step. And then you put those tubes containing the cell extract on top of the sucrose gradient in an ultracentrifuge, and use centrifuge for several hours. And what you're basically doing is forcing those messenger RNAs that are loaded with ribosomes to go down into the gradient, into the sucrose gradient, up to the point where they reach a location within the gradient where they will not continue to move any further. So then they reach kind of an equilibrium, and then at that point you stop the centrifugation. So now you have fractionated the messenger RNAs based on how many ribosomes were associated to them. So messenger RNAs that are located at the bottom of this gradient are the ones that contain the most ribosomes associated to them, whereas messenger RNAs that are in the lighter areas of the gradient contain fewer and fewer ribosomes associated to them. Messenger RNAs that are not being translated, so they don't even have any ribosomes associated to them, will be at the very top. Okay, so now you have fractionated messenger RNAs based on how many ribosomes are associated to them. The next step is then collecting fractions out of this. And how do you do that? Well, one way that you can actually do this, that you can relatively easily accomplish the collection of these different fractions, is by injecting a very viscous substance at the bottom of the tube, so that then you push everything upwards. And then you can put here a seal, and then you can connect a tubing here. That tubing can then allow the solution that will be pushed upwards to go through a detector, which is an ultraviolet detector. So basically you can see the absorbance of the solution as it is passing by. And then you can collect fractions using a fraction collector. So you can collect fractions in different tubes, and you can collect fractions of, say, 1 milliliter or half a milliliter or whatever volumes you want to collect in each tube. So you can program this in the frac collector. And that kind of system is actually shown here. That's what is represented here. So you have an actual pump that will be pumping the solution the high density solution, and typically a solution that is used for these kind of experiments is known as fluorinert FC40. So this is a very viscous solution that will not mix whatsoever with the sucrose that you have in the tube. So it gets injected into the tube, and then the solution will be displaced from bottom to top. So the very first thing that will be coming out of the tube will be whatever is at the very top layer of the tube. So the first fraction to be collected, fraction 1, will contain the things that didn't even enter on the gradient. So it'll be, for instance, the non-translating messenger RNAs. And then the heavier will be in later fractions, so you can collect many different fractions. And the latest fractions will contain the messenger RNAs that have more than a few ribosomes associated to them, something that we will be referring to as polysomes. So keep that in mind. Messenger RNAs that are associated with many different ribosomes at the same time are referred to as polysomes. All right, so then you will have them separated in different tubes, in different fractions. And since you're detecting the UV absorbance of the fractions as they are being collected, you will end up with this kind of profile that is shown here, where you will have absorbance at 254 nanometers, and the first region of the gradient will have a very high absorbance, and that will be things that are free floating, including messenger RNAs that don't have any ribosome associated to them. Then, deeper into the gradient, you will have free small ribosomal subunits, and then the large ribosomal subunit by itself will also be found deeper into the gradient, and then you will have the 80S ribosomes, 40S and 60S ribosomal subunits associated to each other. That's the 80S ribosomal subunit. And then you will have, starting in this region of the gradient, you will have fully associated ribosomes that are in complex with messenger RNA. So here you have one ribosome per messenger RNA. The next peak is 2 ribosomes per messenger RNA. The next peak is 3 ribosomes, 4 ribosomes, and so forth. So somewhere around here, starting at about 3 ribosomes per messenger RNA, you can already call this the polysomal fraction. And the further, the deeper you go in the tube, the more ribosomes you have associated to a given messenger RNA. So the better translated a messenger RNA is, the deeper you will find it in these sucrose gradients. So now what you can do is you collected these fractions, and now you can then, out of every fraction, you can isolate RNA from every fraction. And then you can analyze the RNA that is present in every fraction simply by running an agarose gel using denaturing conditions that will allow the RNA to have a linear shape. This is something that is typically done using formaldehyde to denature tertiary structures so that you can separate those RNAs in the absence of any secondary structures. If you do that, you end up with this kind of profile where the bands that you're going to see are typically those of the 18S ribosomal RNA and the 28S ribosomal RNA, and you may also be able to see free tRNAs. So free tRNAs will be found in the highest region of the sucrose gradient because they have very low density. They are not able to reach the lower densities in the sucrose gradients. So again, this is from top to bottom on those sucrose gradients. So fractions 1 and 2 may actually contain tRNAs, shown here, and fraction 3 may also contain that. Fraction 4 now contains the small ribosomal subunit. Fraction 5 contains now a little bit of the large ribosomal subunit as well as some of the small. Fraction 6 contains a lot of the large ribosomal subunits, and then fraction 7 contains the 80S, meaning large and small ribosomal subunits that are joined together forming the 80S ribosomes. And starting from that point, then the next ribosomes that you have going further down into the gradient are ribosomes that are associated to messenger RNAs. This is 1 ribosome per messenger RNA, 2, 3, 4, and so forth. So this is the profile that you will get in these kind of gradients. The final component of how to analyze these things is when you are questioning now, when you're trying to define where any given messenger RNA is located in these kind of gradients, then you need to do another type of analysis. And that is when you are searching for specific messenger RNAs, then you need to use methods that will allow you to identify that specific messenger RNA that you're searching for. And the method that is typically used for that is RT-PCR or use RT-PCR, depending upon whether you want a quantitative analysis or not. So RT-PCR is reverse transcription PCR amplification. So when you're looking for specific messenger RNAs, you can have specific sets of primers that will target a specific messenger RNA. So if, say, you're searching for the messenger RNA that codes, for instance, for tubulin, just to give an example, you will have then a forward primer and a reverse primer that targets sequences that are specific of tubulin. So the first thing that you will need to do is do reverse transcription. So you will use a reverse primer targeting tubulin, and that will allow you to have now a cDNA molecule for the messenger RNA for tubulin. You can use a messenger RNA specific target to the reverse transcription, or alternatively you can use a poly-T primer, so a primer that contains nothing but a bunch of T's in a row, and that primer will be unspecific. That primer will allow you to reverse transcribe all the messenger RNAs that are polyadenylated in your sample. But most of the time people will actually choose a primer that is specific to their specific target, so in this case tubulin. So that will allow you to have then a cDNA molecule that corresponds to the cDNA of the tubulin transcript, and then you will use another primer to then amplify that cDNA. So now that you have a cDNA molecule here, you can use another primer that will allow you to amplify that segment. And then using those two primers on and on and on in several consecutive cycles of PCR, you can then amplify that specific segment, therefore generating a signal that is specific for the messenger RNA coding for tubulin. So whenever you have the messenger RNA coding for tubulin, you will be able to amplify that PCR product, and that will give you a band. And that band will be visible just as a band on a gel. So wherever you see the band, that indicates that that particular messenger RNA is present in that particular fraction of the sucrose gradient. So the messenger RNA that is being evaluated here in panel C, that particular messenger RNA is found in only the very first few regions of the sucrose gradient, which indicates that the transcript C is not really being very well translated. So messenger RNA C is poorly translated. Now, in contrast, the messenger RNA that is being detected in D is present in regions that span all the way down to polysomal regions. So it's present in monosomes, messenger RNAs that contain 2 ribosomes associated to them, 3 and 4, so up to 4 ribosomes. So the messenger RNA that corresponds to transcript D is better translated than that shown in panel C. And then finally, the messenger RNA that is shown in F is one that spans all the way to the very bottom of the sucrose gradient, which means the messenger RNA identified as F is very well translated. Now, if we were to draw these messenger RNAs and we were to represent it as being associated to ribosomes as shown here, this transcript will be one that is okay translated, this transcript is one that is very poorly translated, and this transcript, the one identified as 3, that will represent a transcript that is really well translated. So 3 will probably correspond to the one represented in F, then 2 will correspond to the one shown in D, And 4 will likely correspond to the one represented in C. So in other words, C, this profile that is shown in C, will correspond to that messenger RNA. It has only one ribosome per messenger RNA, if any at all, whereas the one shown in 3 is the one that is very well translated, so it will correspond to the one represented in F. Okay, so this is the way that you can evaluate translational rates for any given messenger RNA. Now there is one final messenger RNA that is indicated here as not giving a signal at all in the entire gradient. So what could have happened to that particular messenger RNA? Well, one relatively simple answer is that messenger RNA was fairly rapidly degraded. And that could be what happens if you have a microRNA targeting your messenger RNA. The first effect needed by microRNAs is it will halt translation, then it will trigger the deadenylation of the transcript, and finally it will trigger the decapping of the messenger RNA. So altogether, microRNAs will then trigger the decay of the messenger RNA that is being targeted by that particular microRNA. Again, it's a process that takes several different stages, starting with halting translation, but in the end, the final outcome of being targeted by a microRNA is that the messenger RNA will be targeted for degradation. All right, I hope that this gives you a sense of how we can actually assess translational efficiency for any given messenger RNA. This is just one of many methods that allow you to do this. This is a more classical approach. There are other methods that are now very widely used that depend in the direct sequencing of RNAs that are protected by ribosomes. That might actually be the topic of a future episode of the Molecular Cell Biology YouTube channel, but it won't be dealt with in this particular episode. So for now, I hope that this provides you with a sense of different strategies for measuring translational efficiency and how we have become aware of the fact that translation can be very tightly regulated in eukaryotic cells. As always, if you enjoyed this video and found it valuable, give it a thumbs up and subscribe to my YouTube channel. Thank you for watching.